Today we're doing the indicator lab. We'll look at the response to various indicators like universal indicator, phenolphthalein, Phenol Red, and others. The way they change color with various pHs. We have here pH 2, pH 3. These are buffer solutions. They go up to pH 11 and 12. They are standard solutions, and some of them have a color to them. pH 10 here is light blue, and there are other colors as well. They won't affect our results. In the first column, you'll see that we have red litmus paper, and we have 11 pieces of that. You'll start off with 3 strips, and you'll just tear it into 4 pieces per strip. That'll give you 12. You'll do blue litmus paper here, and then pH paper here. You'll only have 1 strip of that, and you'll just tear it into 11 pieces. Red litmus paper comes in a container that looks like this, and the pH paper comes in a container that looks like the one on the right. Most likely your paper will already be taken out of the container and available on the front table. Now we need to put the papers down on your test card. I'll try and do that as quickly as I can here. There's all of the pink, the red litmus, the blue litmus paper, and now we have the pH paper. Next, we're going to put the drops of the standard pH buffer solution. So this is pH 2. We'll put 1 drop in every cell of the test card all the way across. Then we'll do the same with each of the other buffer solutions. So pH 3, 1 drop all the way across. And then we'll do the same thing with all of the other buffer solutions. We'll try and move a little faster here. It doesn't matter what order you do them in. As you find them, you can use them and then put them back in the tub. There's pH 8, pH 7. We're almost done pH 6. And the last one, pH 5. Now, as you can see, the red litmus paper remains red up through about pH 7, and after that it turns blue. The blue litmus has turned red all the way up through about pH 7 and then remains blue. The pH paper starts off at orange, turns yellow, then light green, dark green, and eventually, for the strong bases, it's blue. Now we'll go on and test our solutions. The first solution is universal indicator. You'll notice that universal indicator, as well as some of the others, are marked as flammable. They're mixed with alcohols, which makes them a little bit runnier than water. They don't bead the way water does because they're not as polar as water, so you have to be careful with the alcohol solutions that you get just 1 drop. They also tend to flow out of the tip of the dropper a lot faster, so you really don't need to squeeze them. Next, we'll look at the phenolphthalein solution. You can see the color changes there are quite remarkable for the pH paper and the universal indicator. Nothing happens here with the phenolphthalein until we get down to about pH 8. There's a light pink color and now a much darker pink and really almost a purple. The pH 10 appears to be a little bit darker, and that's because the solution itself is a little bit blue, which has an effect on the color that you see. Next is Phenol Red. The first few are yellow, and then we turn an orange, and then a bright red. Next we have Bromophenol Green. As you can see, it's also yellow. but then it turns green, and then after that all the rest are a quite vibrant blue color. Next we have Bromophenol Blue. Don't confuse this with Bromethymol Blue, which is also in your indicator kit. We're not using that one. We're also not using indigo carmine and possibly some others. And that's just save space. Otherwise, we'd need 2 cards. The next one is Lazarenn. Lazarenn is in methanol, and it's very runny. You almost don't need to squeeze the dropper at all. It literally pours out, almost like the dropper is leaking. You have to be very careful. See, I already spilled a drop. So 1 drop in each one of the cells. Starts off yellow, and then we get to a purple color. And the last one is alizarin yellow R. Even though we spilled some of the alizarin and it ran into that cell, we're not really worried about it because this is going to be the same color anyway. The alizarin yellow R, though, transitions at a different place. As we go down, we can see it's now it's orange there at around 9, and then it's really red, and then a very dark purple color at the very end. Now you should be able to tell the pH of an unknown solution by comparing it to this standard chart that we've just developed. Mrs. Crane's 7-year-old came in and looked at one of these charts, and I asked her the question, if you knew 2 colors, if you knew that the universal indicator was orange and that the Bromophenol Blue was blue, you could read across and see that the pH was 5.